Hello and welcome to the RI Science Podcast. Depending on where you get your podcasts, you may have noticed that this is a video episode. If you're not seeing this video but you'd like to, just head over to our YouTube channel and search The Royal Institution. We're in the midst of a watershed moment for artificial intelligence, and its prevalence in our world is becoming harder to deny. We're diving deeper into how AI is changing the scientific landscape specifically, and this episode we're focusing on a DeepMind machine learning program called AlphaFold. Launched in 2018, AlphaFold has taken the scientific world by storm by addressing the protein folding problem, which has been perplexing scientists since the 1960s. Every protein is made up of a unique chain of amino acids. How this chain folds up determines the 3D structure of the protein, and this structure then determines the protein's job. Proteins are found in every cell of the body, and we have them to thank for everything from making hormones and helping digest our food to maintaining the structure of cells themselves. Knowing the exact 3D structure of a protein can tell us more about their function, and this can contribute to science in loads of different ways, from drug targeting to helping digest plastics. AlphaFold has established the structure of 200 million proteins and has been hailed as the most important achievement in AI ever by Forbes. To discuss the past, present, and future of AlphaFold, as well as the fundamental scientific questions it may or may not be answering, RI event producer Lisa is joined by Dame Janet Thornton. Janet is a champion in the field of bioinformatics and protein structure, and served as the director of the European Bioinformatics Institute, also known as EBI, from 2001 to 2015. She then remained a research group leader there until June of 2023, and also directly interacted with DeepMind when they partnered with EBI to host AlphaFold's protein structure database. Before we dive in, please remember to give this episode a rating and a review to let us know what you think of the new format and to help other people discover the podcast. But for now, let's hand over to Lisa and Janet. Welcome, Janet. Thank you so much for joining me today.
My pleasure.
I'm going to dive right in with the question and just have the overall view. So AlphaFold has been hailed as one of the most important achievements in AI ever. How has it transformed the field of biochemistry?
So it has had a huge impact, actually, absolutely enormous. What AlphaFold does is to predict the three-dimensional structures of proteins. We already had 200,000 structures, so we had quite a lot of structures. But of course, in all the different organisms, there are millions and millions of structures. And what AlphaFold did was to predict rather accurately the structures that are now available, 200 million of them. So we have in the database at the European Bioinformatics Institute, where I work, we have this database with all these structures in that's available for everybody around the world. So all the biochemistry labs where people are interested in one specific protein can now look up the structure of that protein and be fairly confident that it will be correct, even though it's been predicted. It's not an experimental structure.
And in order to understand the significance of what AlphaFold is doing, we need to go down to like a molecular level. So a vast amount of processes going on in our cells are down to proteins. So would you be able to give us a very brief summary of what these biological molecules do and why they're so important?
Okay, so I'll start with what they do. They do everything in the body apart from I would say the DNA that carries the information from one generation to the next. But when you look at a person, what you mostly see is protein. Your hair is protein, your nails is made out of protein, your skin. But internally, they do all the work. So they're like the factory of the body. They're the enzymes that catalyze and do all the chemistry of life. They're the antibodies that provide our defense when we get ill. They're the molecules that transform DNA into proteins. So proteins basically make other proteins so that you can generate a human being or a tree or a whale.
So biology, complicated, messy, and it's hard to sometimes imagine things. So when we're talking about protein, what scales are we working with?
So proteins are very large molecules, but in reality, they are tiny, tiny little molecules. And I think if you imagine your credit card and the depth of a credit card, within that depth, you can fit about 8,000 different protein molecules stacked on top of each other.
Wow.
So they are minuscule.
So there's something called the protein folding problem, which is why we're talking about AlphaFold and what it can do. So we know this isn't a new problem. So how have scientists been attempting to solve it, and what's the history of the problem?
Okay, so the first protein structure was solved in 1963 about, in Cambridge, UK. And even before that structure was solved, people were trying to understand how this linear molecule could fold into 3 dimensions. So if I use my prop, we have
prop.
this excellent prop, a red shoelace, but it's rather like a protein molecule that's unfolded. And the protein folding challenge is to take the structure, the chemical structure. It's made up of our 20 amino acids that make up all proteins. And in the cell, these proteins will fold up naturally with kind of no assistance into small globular structures. And the structures of those determine how they work, how they function. And that's why they're so important. If the protein is unfolded, it doesn't work at all. Also, we know that things like dementia are caused by proteins unfolding in the brain. And so this folding process is really central to life, actually. And without it, there would be no life, as we know it anyway. So the problem, which frankly a young child could understand, is if I give you my red shoelace and tell you what its chemical structure is, are you able to predict how it's going to fold up and the structure that it folds into? And the folding problem actually has 2 aspects. One is how it folds up. So what's the process that goes through that? That was often called the folding problem for obvious reasons. In fact, what AlphaFold does, it doesn't predict the folding itself. What it predicts is the final structure. So you give it the chemical structure of your protein, and it predicts very accurately. So people have been trying to predict protein structures ever since that first structure was solved. And I joined the field in 50 years ago, really, and was fascinated as a physicist. I was fascinated by this problem, because it's such a simple problem. But perhaps I was even more fascinated as we got to know the structures of more proteins. So when I was a PhD student, there were only 20 known protein structures. And when you look at these structures, they're just beautiful. They're very complicated, but they often have symmetry, like flowers, where you get 6 petals or 6 repeats or 10 repeats or 4 repeats, and they bind together so you get multimers, where you get multiple proteins. And they are just beautiful to look at, frankly. And so it was quite a nice challenge to be able to try to predict their structures. So people have been trying, obviously, always using computational approaches because they're big and complicated, to do this prediction. And in the beginning, when I first started, we made quite good progress using the laws of physics and chemistry to not just to predict the final structure, but to predict this folding process. And at one stage, I thought, until we can predict that folding, we aren't going to be able to predict the final structure. And I was wrong, because the AI techniques basically can use the data. They just take the chemical structure and they predict the coordinates. So this it looks rather a mess. I have to say, I have a model here which is far more beautiful, which shows it's actually called a tin barrel, because it's triose isomerase, and it's an enzyme. And its structure is beautiful because it's got 8-fold symmetry, and the many enzymes in the body adopt this structure, and the catalysis, so they're substrates that bind, the small molecules bind in at one end of this barrel. And so it really is a beautiful structure, and it's been used repeatedly during evolution to create new enzymes. And what AlphaFold does is to predict exactly where each atom goes in this structure. And so it's quite an achievement, and one that I had come to the conclusion that would not be solved in my lifetime. And so I was delighted when this, when the AlphaFold news broke.
It's always nice to be proved wrong when it's something like this.
Exactly.
You're not the only person who thinks that structures are interesting, because there are, I think, what's called the Olympics of protein folding.
This was very interesting, actually, because this started, as I said, from about 1970. For many years, people were generating new computer algorithms to predict these coordinates. And every now and then there would be a paper saying, we've solved it, this is it. And 6 months later it proved that they'd not solved it, they'd just done a little bit of the problem. And so John Molt in the States and colleagues decided to get together to have like an Olympics that was called the Comparative Assessment of Structure Prediction, CASP. So every 2 years, there'd be a list of new proteins. So the computing people were given the chemical structure of the protein, and then they had to submit to the lab that was assessing these, the structures. And then people could say whether it was right or wrong. And it was very clear in the beginning that we weren't very good at predicting ab initio, shall we say. We could predict when we knew the structure of a closely related protein. And in evolution, of course, there are lots of those. So that's a whole different area, but it's critical to this prediction. But if it was a completely new protein, then we were no good at all at predicting that. And in fact, I was involved in the sort of prediction game, because it is rather like a game, actually. People, it's a real challenge to try and do this. And I concluded in about 1990 that I didn't know what to do. So I started looking much more at the evolution and the function of the proteins. But a whole group of people carried on trying to improve the prediction. Gradually, the methods being used were refined, and initially there was rapid improvement, but then it slowed down, and the improvement was very slow for probably 10 years before 2018, when AlphaFold, DeepMind, first participated and used their AI technology. I should say other people had used AI in predictions, and that year several groups incorporated different AI methods, and so the prediction improved more than in the previous 10 years, actually.
Wow.
But the big advance came in 2020, 2 years later, when DeepMind had gone back home. They'd redone the whole algorithm. They'd used some of the more modern AI technologies and approaches, and they were remarkably successful, actually. They were the best prediction for over 90% of the structures that were submitted to CASP. I think we have to be fair to all those people, because there are about 100 labs around the world that participate in CASP every 2 years. They had made improvements, and they had made a lot of technical advances about how to frame the problem for the computer. You know, I used neural networks in the early 90s for prediction. So AI, in a sense, had been used right from the very beginning. And use it based on that work, number one, so that how to represent these structures in a way that was computer-friendly and predictable. But number two, there were then 170,000 protein structures. And the wonderful thing about these protein structures is that they'd all been gathered in a database called the Protein Data Bank. And that was set up in 1973. We had, for those 170,000 structures, we knew the chemical composition and we knew the structure. And the data were well organized, they were validated, we were confident about that data. And so, in a sense, DeepMind and AlphaFold, they had what was needed to train their models. And it's just amazing how well it works. They understood about much better than most of the people doing it about the AI and the different AI techniques that are now available that weren't. So there's been huge advances there, where I am no expert, but it certainly has made a difference. So I think it's been a great journey, actually.
We're sat right now in a lecture theatre where a lot of innovations and scientific achievements were made and started right from the beginning. So one of these processes used to discover the structure of proteins was X-ray crystallography, which was pioneered here in the labs at the Royal Institution. It was a father and son team, William Henry Bragg and William Lawrence Bragg, and then further developed by a dream team of crystallographers, Max Perutz, Dorothy Hodgkin, Catherine Lonsdale, Rosalind Franklin, Watson and Crick. And we were having a little chat before we started recording, and you also mentioned Lord David Phillips, who was the head of a lab at Oxford when you were doing your, did you say postdoc there?
Postdoc, yes.
And he actually gathered data here and created the first lysozyme model, which we have in our basement. And so that's quite a nice link. How did we get from that to where we are now with AlphaFold and visualizing these protein structures?
So a lot of work by a lot of crystallographers on solving protein structures. So I moved to Oxford for my postdoc, and the lab there was devoted to protein crystallography. So David Phillips had been here doing the structure of lysozyme, the very first enzyme structures to be solved. And he went to Oxford and set up a lab there, and Dorothy Hodgkin was also part of that. So it was a very important part of the development of crystallography. So in those days, determining the structure of one protein could take 5 or even 10 years. It was a huge task. And the problem was that there was no guarantee of success. It wasn't push a button like with protein sequencers now, We know we can determine them with DNA sequencing. You push the button, put the material in, and out comes the structure, the chemical structure. For crystallography, it was different. And so people worked for certainly the whole of a PhD. Often it would involve 2 or 3 PhD students working on one protein. And in fact, this structure that I showed you before was solved when I was in the Oxford lab.
Oh, wow.
And it was, in those days, it was a long process. Louise Johnson, who was in the lab, was working on another protein. And it was so big that she needed ladders to climb up the Richards box to create this structure, this incredible structure. It was, and then when you'd built this model, you had to physically get the coordinates of each atom, and that had to be typed into the computer. So the opportunity for error was quite large, actually, because
imagine.
Trying to get it all consistent. So it was a long, long process. And that was why people were desperate to be able to predict without going through this experimental problem. Now, things are very different, because now we have electron microscopy that can generate these models without forming crystals. And that has again revolutionised the field.
Let's now move on to the EMBL EBI collaboration. So DeepMind partnered with EMBL EBI, which is the European Bioinformatics Institute branch of the European Molecular Biology Laboratory. So I appreciate EMBL EBI saying that instead, which hosts a database of 200 million protein structures derived from AlphaFold. Could you talk about how this collaboration came about?
EBI is the place in Europe where we gather structures, experimental structures from the European protein structure community. It's called PDBE, PDB Europe. And so, as I said, this is part of the worldwide collaboration. And so we had all the tools to collect structures and to make them available, because EBI is the center of molecular data, really, in Europe, biological molecular data. And so when the AlphaFold story broke, clearly DeepMind were thinking about what to do with all these structures. And they are part of Google, so they are a commercial company, and it wasn't clear at the beginning whether they were going to make these data available or not. And obviously they decided they would like to make their predictions for... Then we thought about the human proteome. But that developed into the the 10 model organisms, and then, in fact, over 1 million different organisms have protein structures in the current version of the AlphaFold database. And so, really all credit to DeepMind that they made these structures available. Samir Valanka at EBI, who heads the PDBE, he was already involved actually in a process to make models available, because we wanted to keep the models separate from the experimental structures. In fact, it's getting more and more blurred, the difference between the two, because even the experimental structures obviously are models in some way. In fact, the head of DeepMind spoke to our Director General, Edith Hurd, in Germany, and obviously discussed also with Ewan Burney about possibly depositing these structures. And they have provided funding to EBI to allow us to create the AlphaFold database, where these 200 million structures are available.
You've touched on it briefly, how the importance of making the database freely accessible.
Most of the data at MLEBI is freely available data. And for the PDB, the experimentalists basically deposit their data in the EBI. Then we make sure the data are well structured and etc. But that's fairly automated now. And then we create a resource that people can go and download. So in fact, you could download 200 million structures if you wanted to, but that's probably not the way to do it. But you can do, the ones that you want to look at. So the process is, deposit, curate, annotate, make available. So that it's sort of like a virtuous circle. And that circle has been running now for almost 50 years. And it's only because of those data which have been collected and made freely available that it was possible for AlphaFold to do, because for any AI, the training data are critical.
Yeah. Is AlphaFold the only one of its kind? Is it kind of monopolizing the field of protein folding?
I would say it is monopolizing at the moment. When people talk about using models, they're nearly always the AlphaFold models because they are still the best models.
So let's move on to future developments and applications.
Yes.
So even whilst the team have been researching this topic, there have been developments. So recently, we've seen two recent developments. The first is AlphaMissense, launched in September of this year, which is using amino acid sequences to determine something entirely different to protein structures. Do you have an idea of what that involves and what that might mean for the future and its impact?
So this is a little bit like the protein folding problem. For many years, people have wanted to predict the effect of variants, genetic variants in your DNA that translate into changes in one of the amino acids or more in a protein sequence. And that in turn may change the structure that the protein adopts. And so the key when I think the clearest, there are several examples. In cancer, this is the effect of mutations, these variants in your DNA that cause cancer cells to develop. Also in developmental disorders in children who, for whatever reason, have a variant in their DNA that causes development to stall or to be a problem in development. And so predicting the effects of these variants, they're variants in the DNA that lead to variants in the proteins, is really important for any genetically inherited disease and for cancer. And so the idea was to, in fact, the original folding, if you use the folder structures that are in the PDB database, PDB AlphaFold database, they are not very good at predicting whether one of these variants is benign, i.e. doesn't have any effect, or it's pathogenic. Because if you have a child with a developmental disorder, you can sequence their genome 3 times 10 to the 9 bases, and then you have to find the 1 or 2 variants that might be causing the developmental disorder. And so identifying variants and saying, is this a benign or a pathogenic variant, is very important. So this is what AlphaMissense tries to do. It uses additional data. So it uses the structural data, their predictions, and it also uses data. So there are databases that tell you this variant is associated with this disease. So you have data on variants which are pathogenic and variants which are benign. That said, those data are not as large in some ways as the whole of the protein data bank, and their interpretation is difficult. We were talking earlier about how biology is messy. Mm-hmm. This is one of the cases. It's quite difficult, actually, to predict. Some variants are very easy to predict, and you don't need AlphaFold. You can just look at it almost and say, that's a bad one. Others look perfectly benign, and yet they'll be bad. Others look disastrous, and they have no effect. So it remains difficult, even with alpha missense, to predict the effects of those variants. And so a colleague said to me, this isn't an AlphaFold moment. It wasn't that there was this massive increase. And I think that's explained. It was better, but not a step change. Mm. And so I think that reflects the state of the data and this messiness that we talked about.
People do argue that biology is the hardest of the subjects.
Yes, well, it certainly is. Yeah.
And at the time of recording this, there was an announcement a couple of weeks ago, at the end of October, for AlphaFold latest. How does this differ from the previous version of AlphaFold?
In the original AlphaFold, they have no ligands. So ligands are the small molecules. So for enzymes, they're the molecules that are acted on, catalyzed by the enzyme. But almost all proteins have small molecules, cofactors, coenzymes, metal ions, or metabolic small molecules like ATP bound to them. And in these models that AlphaFold produced, there were no ligands at all, which makes no physical sense, because if you have a protein, many proteins, not this one, but many proteins have metal ions that stabilize them. So you have a zinc or a magnesium, and that holds the protein or helps to hold the protein together. But they weren't there in the AlphaFold structures, and yet they predicted the right structures as if they were there. And so this just shows that it's not based on physics and chemistry, it's based on copying what they've already seen in the PDB in a very sophisticated way, but nevertheless, that's what it's based on. And so now they're moving into looking at ligands. And one of the most important things proteins do is to interact with DNA. And so many of these, not that many, but several thousand or more, have DNA bound to the protein. And you want to be able to model that. They also, you didn't mention AlphaFold multimer. Quite a lot of proteins in nature exist as dimers, where you get two identical proteins that form a complex, or tetramers, or even higher order complexes. And so AlphaFold multimer is really quite good at predicting these complexes. But in that press release, it was about the ligands and the DNA. Now, it's interesting. I really don't know how easy it's going to be able to predict the ligands, about the small ligands, because, as I said, the predicting protein structures is the ideal AI problem, because you've got 170,000 protein structures, all made up of identical amino acids. So although they're different because of the different amino acids, they're still the same. So you can, every protein has its arginines, its the different amino acids in there. The ligands are different. They're much more complicated. Even though they're tiny, they're actually not that well behaved as proteins, which is surprising because they really, compared with 3,000 atoms in a protein, these small molecules often only have 30 or 40 atoms, and yet they are quite difficult and they're all different. And I'm sure that there will be a way around it. And of course, they can begin to incorporate all the rules and all the knowledge of small molecules that are in another database called CCDC, where you can abstract basically organic chemistry rules from there. So I don't know whether it's going to be as easy, but of course that has commercial implications as well. Because if you can predict the ligands, you can begin to design drugs, basically.
Wow. And that would be amazing.
Yes, because, well, also, surprisingly, human beings have just over 20,000 different proteins. Although we know what some of them do in great detail, there are a lot that we still don't really know what they do. We know sort of what they do, but we don't know the exact substrate that they act on. And so that's a big problem that is AI tractable. I don't know how successful it will be.
You've already spoken about the profound impact that AlphaFold has and can continue to have. Do you have any concerns about AI in your field?
As always, with all scientific, if you like, discoveries or progress in technology, it can be used for good or evil. And of course, with this, one of the next big challenges that I haven't described is potentially protein design and possibly virus design. And you could imagine that using these technologies, people could design really nasty viruses. And so, in fact, AlphaFold database doesn't include any virus data for that reason. In my field of protein structures, I don't have concerns. Perhaps in the broader field, because these new AI methods are really immensely powerful if and only if you have good data. And one of the areas I think where it will be really important is improving data-driven understanding of medical diseases. Because obviously there is huge amounts of medical data. There are big efforts to coordinate collection, obviously making sure that it is secure and people are not, the need to keep people's identity private. If that can be done, and I think it can be done effectively and efficiently, then I see over the next 10 and 20 years, these approaches can revolutionize the practice of medicine, actually, by using the data to diagnose what's wrong and to make a prognosis of how that disease is going to develop. So that is a huge area of interest. There are concerns as there in there because it involves individuals and people's life and health. And so that has to be treated with immense caution. But at the same time, the opportunities to understand and be more predictive are vast. And so overall, I'm very positive about it and think we should push forward with that quite as quickly as we can, because it will really mean that diseases can be treated more effectively.
To wrap up this whole amazing conversation. Thank you so much. I'm basically going to ask you what seems like a simple question, but has AlphaFold solved the protein folding problem?
So I'll give you a sort of a weasel answer to that.
Absolutely fine.
The protein folding problem, as I understand it, is describing how the protein changes from this extended form into its folded structure. AlphaFold has not solved that problem. All it does is to give you those coordinates. So in my terminology, AlphaFold has not solved the protein folding problem. It solved the protein folded problem. I mean, the problem always with AI is that you don't really understand why it works. Frankly, I think of AlphaFold as a very sophisticated cut and paste algorithm. So it learns from every single little bit of the protein structure how best to put it together. But we don't learn more about the physics and the chemistry, and this comes down to the dynamics of the structures that we've talked of before. The other big thing is, of course, that these structures, tiny though they are actually flexible. And these things move. They're dynamic structures. They breathe, literally. And sometimes the function depends on that breathing. It depends on these slight movements. Or actually, sometimes large movements. Sometimes they do big things like that. And so knowing the structure is beautiful. I delight in it. But actually, what we want to do is to understand how they work. And to understand how they work, we have to understand the function. And that function often involves this dynamic aspect. And that has yet to be. It's not been really addressed by what AlphaFold has done so far. So it's just the beginning in many ways.
Excellent. Well, there's even more to come. Janet, that was insightful and extremely interesting. We could chat all day about it, I'm sure,
yes,
but we don't have time. So thank you very much for joining us here at the Royal Institution.
Thank you for the question.
That's it for this episode. Thanks for listening. We hope you learned as much as we did from Janet and might even be tempted to head to the AlphaFold database and find your own favourite protein. If you want to explore more AI, make sure to tune in to the Christmas lectures on the 26th, 27th and 28th of December on BBC 4. And if you're listening to this after, find them on BBC iPlayer if you're in the UK or on our YouTube channel if you're anywhere else. In the meantime, head to rigb.org to book tickets for our upcoming talks from more amazing speakers.